This method makes it possible to identify ubiquitination of EYFP-tagged CENP-A protein. It can also be applied to human CENP-A protein with different tags and other centromere Kitagawa proteins.
The main advantage of this APMS strategy is that it can be used to identify EYFP-CENP-A K124R ubiquity sites with crucial biological significance. It could be extended to investigate the post-translational modifications of a wide range of functional proteins.
Begin by preparing protein A beads bound with anti-GFP antibody. Wash 25 microliters of protein A beads per immunoprecipitation reaction with buffer A1 at least 3 times to remove the ethanol. Then make a 50% bead solution with buffer A1. Add 2 microliters of anti-GFP antibody to the beads. Then add buffer A1 to 20 times the net bead volume. Perform end-to-end rotation at 4 degrees Celsius for 4 to 18 hours. The optimal duration for the rotation must be determined empirically based on the efficiency of the immunoprecipitation. After the rotation, centrifuge the beads at 100 times G for 1 minute and remove the unbound supernatant. Add buffer A1 to make a 50% solution of beads and use 25 microliters of this solution for each immunoprecipitation reaction. To perform the immunoprecipitation, lyse cells in buffer A1 via sonication and freeze thaw. Measure protein concentrations and normalize the protein amounts among the different IP samples.